All right. Thanks, Jeremiah. Good afternoon, everyone. My name is Michael, and today we're going to be talking about T cells, their functions, and how do you track them. Hold on. Okay, cool. All right. So in this presentation, we're going to talk about a little bit about the innate immune system. We're going to then go on to the adaptive immune system. We're going to talk about the discovery of T cells, the journey of the T cell maturation. We're then going to focus on helper T cells, cytotoxic, regulatory, natural killer, memory, and effector T cell functions. So the innate immune system is composed of different leukocytes. These leukocytes are neutrophils, monocytes, eosinophils, basophils, and natural killer cells. The roles in the body for basophils and eosinophils is more active for allergies. Neutrophils tend to be our first responders, and they are the most abundant. Natural killer cells are large lymphocytes that don't really need an activation to locate infected cells and cause them to apoptose. Macrophage are kind of the cleanup of cell debris, and they can also be antigen presenting cells. And dendritic cells main role in the immune system is acting as an antigen presenting cell. So I'm going to briefly talk about the role of the innate immune system. If you want more in-depth information of how it's uptaken and its movement and signaling throughout the body, Josh presented a very in-depth presentation 2 weeks ago. So imagine that you get a cut, and in this cut, bacteria are able to infiltrate your tissue. Your tissue resident cells will then start to send out cytokines. These cytokines will then call immune systems through chemotaxis to bring the immune cells into that area so that they can start fighting the infection. And their role and goal is to control the spread of the pathogen and hopefully eliminate it. But what happens when the innate immune system can't stop the spread? And so another role of the dendritic cell is linking our adaptive immune system. So we have something called acquired immunity. And these are our specialized cells that are able to distinguish and develop a memory against specific pathogens. This is the goal of vaccines. They want to activate the adaptive immune system. And it has two pathways it can go through. It has humoral immunity, and this is the activation of B cells. For B cells, once activated through its receptor, which is specific to a specific pathogen, it starts to produce antibodies. And this antibodies can bind to several pathogens, making it easier for uptake. So this is an extracellular response. A second pathway is called cell-mediated immunity. In cell-mediated immunity, we activate T cells. T cells can then go and determine cells that have been infected. It then is able to force these cells through apoptosis. So this is an intracellular response. So we have to give credit to where credit is due. Dr. Miller, who was born in 1931 in France, his family moved to Shanghai during World War II, and his sister died there of tuberculosis. He remembers that the doctors told his family that they did not understand why her body reacted the way it did. And so this kind of was his inspiration to go into medical research. In 1963, he did some experiments where he took the thymus out of mice, and he showed that the mice without thymus were more susceptible to diseases. Further, he then implanted organs from other mice and showed that these mice would not reject these implants. When he put the thymus back into the mice, he showed that the implants would start to be rejected. His discovery found that thymus has a role. Previously, it was thought that thymus had no role and it was just a waste of an organ. He then in 1968 proved that T cells help B cells generate antibodies. This discovery was the first signal. This was the first time to show signaling is very important in fighting diseases. Thanks to him, we now have the field of T cell biology, which helps in our understanding of vaccinations, autoimmune disease, and cancer. So T cells and all leukocytes are produced in a primary lymphoid organ known as the bone marrow. In the bone marrow, we have lymphocytes, and these lymphocytes can produce into natural killer cells, T cells, and B cells. We're not going to call these lymphocytes T cell precursors. The thymus will send out chemokine signals known as thymosin, thymotaxin, and thymopoietin to call the T cell precursors into the thymus. The thymus is another primary lymphoid organ, and what it does is with those chemokines that it sends out, it causes the T cell precursors to start expressing CD8, CD3, and CD4. It also causes the T cell precursor to start generating what's called RAS1 and RAS2. RAS1 and RAS2 are responsible for rearranging TCR genes. In this rearrangement, we get T cell receptors that are specific to every T cell in the body. So it's unique. It's unique in its T cell receptor thanks to RAS1 and RAS2. This immature thymocyte will then interact with thymic epithelial cells and will undergo what is called the first step of its maturation, which is called positive selection. Here, we focus on CD8 and CD4's binding to MHC molecule. If it doesn't bind, it will undergo apoptosis. If it does bind the MHC molecule, it'll move forward into the next step. In the next step, which is called negative selection, we now focus on our T cell receptor. It is very important that the T cell receptor does not bind to the MHC and MHC2 molecule. If the T cell receptor binds to the MHC and MHC molecules, it will undergo apoptosis. If it doesn't undergo apoptosis, these cells will then start attacking your own cells and is responsible for autoimmune diseases. If it doesn't bind, it moves forward and through random selection begins to upregulate either CD4 or CD8. Whichever is upregulated, the other is stopped being produced. For example, in our CD4 positive upregulated cells, we now have naive helper T cells. For our upregulation in CD8 positive, we have naive cytotoxic T cells. And so from the thymus, once it leaves as naive T cells, it moves on to a secondary lymphoid organ. These secondary lymphoid organs are spleen and your lymph nodes. Here, they await activation to become effector cells. Cytotoxic T cells can be activated by major histocompatibility complex 1. These molecules are present on all nucleated cells. In general, your cytotoxic T cells should not bind to this MHC1 molecule. But if your cell is infected by a virus, the virus will start changing the genes in your cell to start producing viral proteins. When it starts producing viral proteins, your cell will start collecting some of the viral proteins and is able to then put it onto an MHC1 molecule that is produced at its surface. If you find the correct T cell, if the correct T cell receptor binds this MHC1 molecule along with CD8, it activates the cytotoxic T cell to apoptose that cell. For MHC2 molecules, these are only on antigen-presenting cells, for example, dendritic cells, macrophage, and B cells. When these antigen-presenting cells endocytose a virus, it will then reduce them into smaller peptides with proteases. And it will then merge with an MHC2 and push, and then attach itself to the surface of the MHC2. If you find the correct helper T cell that can bind to this now antigen of the virus with CD4, it activates the helper T cell. And now you have mature helper and cytotoxic T cells. So before we move any further, I want to explain how you can track and distinguish T cells from other cells. And this is how to track all cells in general. We look at the cluster differentiation markers. For example, helper T cells are CD4 positive. The problem is CD4 positive cells are also monocytes, macrophages, and dendritic cells. Cytotoxic T cells are CD8 positive, but CD8 positive cells are also found on natural killer cells, thymic cells, and dendritic cells. For T regulatory cells, these are also CD4 positive, and to distinguish them from helper T cells, we can track them by looking at the FOXP3 expression. For natural killer T cells, which is a rare subset of T cells, they express CD161. But CD161 is also expressed on natural killer cells. I'm going to talk about three more CD markers that aren't very helpful in distinguishing T cells. CD45R is expressed on all lymphocytes. CD2, which helps in adhesion, is expressed on all leukocytes. And LFA1, which helps T cells migrate from the bloodstream into an inflamed tissue, is present on all leukocytes as well. So you're probably wondering, how do we distinguish T cells from other cells? And so the main CD we look at is the TCR-CD3 complex. We can use CD3, which is specific to only T cells, and combine them and label both CD3 and CD4s, which will give you CD3 positive and CD4 positive, to know that those are helper T cells. If you have CD3 positive and CD8 positive, you have cytotoxic T cells. If they're CD3 negative and CD8 positive, then they're probably a dendritic cell. But you would have to add other markers to be able to know that for sure. You have other CD markers like CD25, which interacts with interleukin-2. You have CD28, which interacts with B7 on APCs. You have CD44, which is for activated T cells, and CD62L, which tends to be for naive and memory cells. You can you can label your T cells depending on what organ or what area you take it from to kind of determine the whole. You can track your T cells throughout every step of the process. So, for example, if you want to track T cells if you want to track T cells in the thymus, you would want to incubate thymus T cells with CD44 positive. You would want to track them with CD44, CD25, CD8, and CD4. Your precursor T cells will be CD44 positive, CD25 negative, CD8 negative, and CD4 negative. Once it enters the thymus, it will then be CD44 negative, CD25 positive, and still CD8 negative and CD4 negative. Once it gets to the selection process of the thymus, it becomes CD44 negative, CD25 positive, and CD8 positive and CD4 positive. This is the only time when T cells are CD8 and CD4 positive. Once it leaves the thymus, it becomes CD4 positive, CD25 negative, CD44 negative, and CD62L positive. So this is how you can track your T cell through every process of the thymus. Once it's outside, like I said, it's a naive T cell, and you can track it by central memory and effector memory T cells. So once these naive T cells are activated, they tend to upregulate CD44 positive cells, CD markers, and they downregulate CD62L. But for central memory, they tend to still have a high regulation of CD62L and CCR7 positive. For effector memory T cells, it has a CD60L low expression and a CCR7 negative. So you can track your T cells from the thymus all the way till it's a naive, all the way to activation, and then when it's a memory T cell.
Michael?
Yes.
Can you explain the difference between effector memory and central memory T cells?
I was going to explain that when I get to the when I talk more about effector and central memories. Do you still want the answer now?
No, I can wait.
Okay. So for activation of helper T cells, I mentioned that it was very simple. You just have it to attach it to MHC2 molecule. But for naive helper T cells, it actually is a little bit more complicated. These are mainly activated by dendritic cells. And the way it works is you actually need 3 signals to activate a naive helper T cell. The first one being the CD4 and T cell receptor binding to the MHC and antigen molecules. It needs the secondary signal is B7, which is CD80 and 86 binding to CD28. And its third signal is when APCs express interleukin 1 to bind to a receptor in the T cell. With those 3 signals, the naive T cell is fully activated and begins expressing cytokines. Depending on the cytokines that it produces, we can then determine whether it's a T helper 1, T helper 2, T helper 17, or T regulatory cell. For each T helper cell, if it's an interleukin 12, you have Th1, interleukin 4 is Th2, and so on. T helper 1s are very important for cell-mediated response. It can still produce, it can still help in producing antibodies, but it tends to lean more towards activating cytotoxic T cells. We can distinguish them by their production of IF and gamma, STAT4 and TBET. STAT4 and TBET help Th1 cells produce IF and gamma and TNF alpha, which are inflammatory cytokines. For Th2, we can distinguish them by interleukin 4, STAT6, and GATA3. STAT6 and GATA3 are very important for helping promote expression of interleukin 4 and interleukin 5, and those are very important in activating B cells. So Th2 tends to be more of a humoral response type of response. And they tend to downregulate or hinder Th1 cytokines. So they're more anti-inflammatory. Th17 is what I can tell you about it is it's pro-inflammatory. It's inflammatory as well, just like Th1. There's a lot of confusion of how it works because it works a lot with T regulatory cells, which are supposed to be suppressors. And in terms of inflammatory, interleukin 7, you can distinguish these Th17 by interleukin 17, STAT3, and Rho gamma T. Interleukin 17 can help promote the expression of TNF alpha, and STAT3 and Rho gamma 3 are responsible for the upregulating MHC molecules. So, and for T regulatory cells, these are produced by TGFB, and these are suppressor T cells, which I will talk about later on. But you can find them by the expression of TGF beta and FOXP3. So now for cytotoxic T cells, we talked about how helper T cells are activated by antigen presenting cells. We now know that these helper T cells are T helper 1 cells. When the T helper 1 cells are activated, they release cytokines called interleukin 2. Interleukin 2, when it can activate cytotoxic T cell when it binds to an APC expressing MHC1 molecule. This activation allows it to go out and look for infected cells that can then force it to apoptosis. It forces apoptosis by releasing perforin. These perforin can develop holes in the infected cells to allow granzymes and granulocytes to go in and start chewing up the viral proteins and other proteins inside that cell, promoting apoptosis. For regulatory T cells, like I said earlier, they're suppressor T cells. They play a critical role in autoimmunity. So in the process of the thymus is very important in deleting T cells that can attack your own cells. But in some cases, these cells are able to escape the thymus. It is the role of the T regulatory cell to go and make sure that those cells are apoptosed. It also works in suppressing different cells that are pro-inflammatory. So it can cause endothelial cells to reduce its adhesiveness to leukocytes and prevent immune cells from entering an inflamed site. It can release TGF beta to cause dendritic cells to stop producing MHC2 molecules and causes dendritic cells to stop migrating to the lymph node. It can suppress the T cells, specifically Th1 and Th17 cells, from proliferating. It stops this by preventing dendritic cells from connecting to T cells by PD-1. And lastly, it can force macrophages, which are activated M1 macrophage, which can release interleukin-1 beta, which is responsible for activating T lymphocytes, to change from this phenotype to M2 phenotype. M2 phenotype macrophage tend to release interleukin-10, and interleukin-10 is responsible for downregulating MHC2 molecules and is more of a relaxing macrophage. So this is why it is very important in cancer to recruit T regulatory cells, and why cancer immunotherapy focuses on removing T regulatory cells so that we can reactivate an inflammatory or a Th1 response into cancer cells. Now, natural killer T cells are actually not a part of the adaptive immune system. They are a part of the innate immune system. Natural killer T cells tend to be more important in fighting against tumor cells. Natural killer T cells are like T cells because they have a T cell receptor, and they're like natural killer cells because they have a CD161 marker. Natural killer T cell receptors bind to CD1D, which is an MHC type molecule. It can then directly kill tumor cells, or it can activate natural killer cells to kill tumor cells. Natural killer T cells can also be activated by antigen presenting cells, and then can kill tumor cells by activating natural killer cells. And this is a third way that it helps in reducing tumor cells. And this is still hypothesized, but what they believe is it interacts with tumor-associated macrophages and suppresses angiogenesis. Tumor-associated macrophages tend to produce angiogenic factors, which allow for the production of blood vessels. With natural killer T cells, it can suppress these factors and reduce any nutrition going into tumor environments. So now for memory and effector T cells. There are 4 different models for understanding memory and effector T cells. What they believe is in model 1, naive T cells are activated by an antigen. And when it's activated by an antigen, it can produce an effector cell or a memory cell. In all cases, the effector cells will die out after getting rid of the pathogen, and the memory cell will remain. In model 2, the naive T cell is activated by an antigen to make effector cells. Once the effector cells are done clearing out the pathogens, those that survive will then become memory cells. In model 3, it's thought that early effector cells will produce central memory cells, and late effector cells will produce effector memory cells, which can become central memory cells. To distinguish the two, we look at CCR7 and CD62L. Central memory cells tend to reside in lymphoid tissues. And effector memories, which are CCR7 negative and CD62L low, tend to reside in the site where the infection began and is able to circulate in the bloodstream. This is why this is why we have a stronger immune response, because these guys are already waiting and ready to react against a second infection of the same pathogen. And it's actually central memory and effector memory cells are less regulated than naive T cells. Remember, naive T cells needs 3 signals to be activated. Central memory and effector memory T cells can be activated just by the MHC molecule. And that's why our a secondary encounter with a pathogen is a lot weaker and we have a stronger immune response against it. Model 4 is several exposures to antigen begins to decrease the effect of effector cells. And in each case, as it gets weaker, it's still able to reduce memory cells, but at the it becomes non-functional over time, and those effector cells just die out. Now, the most widely accepted models are Model 2 and Model 3. So I am now going to move into at least one paper, and I found this paper very interesting because they looked at 2 different nanoparticles. They looked at lipids and PLGA nanoparticle. Their goal in this paper was to show and determine that slow release can activate a stronger adaptive immune response. It can activate a stronger humoral response and a memory response to the antigens inside of it. So what they did was for the lipid, they used PC and cholesterol at 2:1, and they looked at PLGA at a 50-50 co-block polymer, and they made both these nanoparticles at 200 nanometers in size. Their antigen that they looked at is ovalbumin. In their first graph, they tested, they incubated their particles in PBS to show that the PLGA nanoparticle releases slower than the liposome. They then showed that mice injected subcutaneously with the PLGA and liposome at 100 microgram encapsulated ovalbumin showed that the PLGA nanoparticle, which had a slower release, develop a higher amount of antibody production against ovalbumin. They then took the spleens of these cells and made them into single cell suspension and showed that the PLGA nanoparticles had a higher IF and gamma production, which is very important for Th1 type molecules. And then in the next thing they did was they tested different molecular weights of PLGA still at 50-50 to show that a slower release of PLGA develop a higher amount of antibody production than PLGA that releases it faster. They then use PLGA slow for all of the other experiments. So here's where for me, it got very exciting. And what they did here is they looked at PLGA versus liposome, alum, a known adjuvant for vaccines, and PBS as a control. They labeled the spleen cells with CD8 and IFN gamma, which is an intercellular marker. They showed that PLGA produced the most CD8 positive and IFN gamma producing cells. They then gated these cells to show which ones were activated by looking at CD44, and they made sure that it was antigen specific by looking at a tetramer peptide that binds to the T cell receptor. And again, they showed that mice injected with PLGA, which released slower, developed a stronger memory response that is specific to the ovalbumin, was a lot higher than liposome, alum, and of course PBS injected mice. So I hope that in this talk we learned. You guys, go ahead.
Go ahead.
So I hope that in this presentation you learn how important T cells are in autoimmune, in cancer immunotherapy, and vaccines. There's still a lot being discovered about T cells. They're still determining new subset of T cells found in different organs. They still are trying to determine how the selection of T cells occur. For example, how does it pick T helper 1 versus T helper 2? And trying to understand the memory time frame. Why do some vaccines only last 2 or 3 years and other vaccines can last up to 10 years? And understanding all this can help in improving these therapies. I will now open the floor to any questions. And thank you for listening.
Okay, that was very good. I have a couple questions. In
Yes.
the study that you presented with a PLJ, what was the humoral response? What were the antibody titers relative to albumin?
Oh, I don't think they injected just oval albumin. I don't know if they use that as control. This was published in Biomaterials. I don't remember seeing, I just remember them comparing it to PLGA liposome and alum.
Seems like an obvious control.
Yeah, no, I do agree.
But
Yeah,
did they measure antibodies at all?
yeah, they did. Right here is the antibodies against oval albumin.
So, I mean, and compared to alum as the adjuvant.
Yeah, yes, it did. I left that out due to it not really fitting into the slides. What they show with alum actually is that alum does produce more antibodies compared to PLGA. But I can't remember at what time point they checked it out. I'm not sure if they would have went for longer, how that would have changed. But at the time point they chose, alum had a higher antibody production.
Okay. Okay. And then my second question is, I'm still not 100% clear on the difference between the effector memory cells and the other one. Can you?
All right. So it gets very complicated. If you're just talking about central memory and effector memory, the difference is central memory tend to reside in your lymph node. And CCR7 positive and CD60L high are responsible for bringing it back into the lymph organ. Effector memory cells are more, since they're CCR negative and CD62L low, they tend to reside in the tissues where the infection occurred, and they are also able to migrate and stay in the circulatory system, basically monitoring and being prepared if there's a second infection. Is that what you're asking, and does that answer your question?
I mean, it answers, but I'm not sure what the purpose would be of having a central memory cell that just lives in the lymphatic tissue. I mean, do they like train new effector memory cells that go out, or
Well, yeah.
differentiate them, or
so
it looks like it looks like it goes the other way. It looks like everything just becomes a central memory lymphoid, central memory cell in the lymph tissue.
Exactly. So basically, if the effective memory tends to patrol, but eventually, if it doesn't encounter that pathogen for a long time, it then can resort back into a central memory lymphoid tissue. It will then reside there, and if we encounter that pathogen in the future, again, the APCs can come down and reactivate this to again produce into effector cells, and then again, some of them will become memory cells. If we are infected earlier, your effector memory can help because it prevents the antigen presenting cells from traveling all the way to the lymph node and can cause a response right away near the inflamed tissue or wherever it's infected.
Okay. All right, those are my questions. If anybody else has any, go ahead.
Michael, whenever you were looking at stuff about T cells, did you ever find anything about DNA methylation?
I didn't really see anything about DNA methylation. I know that in some studies they look at. I don't know anything about that. What do you know? What is that for?
Whenever I was looking up stuff for my talk, I kind of like got off on a sidetrack and went down that path a little bit, but usually it serves to inhibit gene expression, but then there are some papers saying that it can be used to enhance T cell regulation. I was just curious.
Yeah, no, I did not encounter that. But I'll write that down to look into it further.
Hey, Michael, I had just kind of a silly question. But so does T cell stand for thymus cell?
Yes, T cells stand for
What does
thymus.
B cell stand for?
Bone marrow.
Bone marrow. Okay, interesting. I did not know
It,
that.
it depends on who you ask. Some people do consider it to stand for bone marrow, but others will say that it comes from I forget the name of it, but some people will say it's a different name, but most people will commonly associate it with it coming from the bone marrow and being matured in the bone marrow. So it's a bone marrow cell.
Very cool. Thank you.
No problem.
Michael,
Yes.
so I have a question regarding the major histocompatibility complex, I guess. So what are the difference between this type 1 and type 2 major histocompatibility complex during the infection? Like which
So,
one is changed? Like, yeah.
so for what I mentioned, MHC1 molecules tend to be on all cells, and they tend to produce a self antigen, and it is to let the body know that it's healthy. But in case of a virus infecting that cell, the viral proteins can interact with the MHC1 molecule, and when it changes it and starts presenting the viral protein instead, it allows your T cell, which normally does not bind the MHC1 molecule, to bind it. And these tend to be for cytotoxic T cells. It's kind of what helps activate it. MHC2 molecules are only present in APCs, and so um, these are produced when your APCs endocytose a virus. It endocytoses it and causes it to with protease make it smaller peptides. It then can present that viral peptide onto its surface to activate helper T cells. So MHC2 is responsible for activating helper T cells, and MHC1 is responsible for producing cytotoxic T cells. Does that answer your question?
Yeah, yeah. Thank you.
Of course.
Hi, Michael. I had a quick question. It's more of a broad question, but what are the downsides to like immunotherapy? Is it just like we don't know as much about it, or it's harder to like figure out what is being like what is controlling the response from certain diseases or things like that? Because this seems like, every disease would create a response in the body, right? So this seems like a foolproof way to target every disease, right?
Well, yeah. So I see what you're saying, and the problem is, the cytotoxic T cells and the helper T cells, they're all cells still. So in some diseases, they're able to target those cells, and so it kind of reduces the response. And sometimes, it's a problem that they don't really understand. But T regulatory cells can also prevent and help pathogens spread further. So regulatory T cells are good because they suppress the immune system, and they stop your immune system from going out of whack and producing so many different cytokines that can cause apoptosis and cause death. So regulatory T cells are good, but sometimes they can be harmful. It's basically the immune system trying to balance itself out to make sure that it has a good response, but at the same time, not too strong. And so I think sometimes if you know what type of disease you're aiming for, and for example, regulatory T cells are hurting the response of your immune system, you can then find ways to target the regulatory T cells to downregulate them and produce a stronger immune response towards that disease, which in some cases in literature they have been doing. But some people have showed that also regulatory T cells, if you downregulate it, it can actually make the infection worse. So it's still a lot that we don't understand of how we can manipulate what we are trying to activate. And I think that's kind of the problem, is that we don't fully understand everything yet.
Yeah. Is that why combination therapies, like putting two different drugs together, is preferred?
I mean that can help. You know, and you mentioned immunotherapy. So like for that, if you can you combine two different therapy, you combine it with a drug, you can make a stronger response by knowing what you're targeting.
Yeah.
Yeah.
Okay. Very cool talk. Thank you.
Hey, Michael. I have kind of a quick question. I have so much questions right now regarding T cells and like the immuno kind of things. So you talk about PSGA and the liposome stuff that they talk about a research, and they prove that the alum is more effective.
In terms of antibody production, it seems like alum was more had a greater response.
Yeah. Yes.
In terms of a cytotoxic and memory response, PLGA was better.
Okay, and this may be kind of out of question. So, when they talk about the liposome, so the major problem is to be addressed with the liposomal vaccines is the direction of their, like, we have to deliver them to a specific site. So when we deliver them to the specific site, so we need to functionalize the surface of liposome in terms of the like incompatible with the like T cells or those receptors. So how can we functionalize these liposome surfaces to direct them towards the specific sites, like
Mhm.
to increase the immunity? Do you have any idea? So it's like major problems to be addressed.
Yeah, no, I would think that you would probably want to target maybe like an APC
Yeah,
rather than a T cell.
yeah,
But I'm not entirely sure how that mechanism would work.
yeah. So,
Um,
yes, it's kind of a major problem, I think. So we have to functionalize the surface to deliver the specific sites. Yes, okay.
and I think if you could find a way to keep it longer in where you inject it, because I think with liposomes they tend to move to the circulatory system rather quickly.
Yes,
If you can develop adhesion molecules to keep it in like your skin tissues instead, I think that having it stayed there and giving antigen presenting cells a higher chance of recognizing it would lead to a stronger immune response. That's the reason why PLGA nanoparticles, I think, or microparticles at least, since they kind of embed like an implant and then slowly release, is why we see this improved or enhanced immune response. So I
yes,
think you know, it just depends, and there's also toll-like receptors and all these other things that you can target as well that could probably enhance your immune response as well.
yeah, it makes sense. Thank you, Micah, for the clarification.
Of course.
Okay, if there are no more questions, then let's all give claps to Michael for a very good talk. Yay! Right, clap, 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 clap. All right, that was very comprehensive, and that was good. I'm looking forward to B cells being presented and all that jazz when you do it next. And yeah, then we've got our regular meeting on Wednesday. Who's giving that?
It's me, Tahmid.
Ah, it's you, Tamid. All right. So that's the plan. Okay. Thanks, everybody. And I'll see you guys in a couple days. Bye.
Thank you.
Bye. Good job.
Bye.
Thanks, Michael.